Hello, my name is Joshua Mugabi. I welcome you to Enzymes Part 2. Bingo! Without wasting any of our time, let's go directly to our quiz. A. An enzyme produced by small intestine that converts lactose to glucose and galactose during the process of digestion. A. Lactose. B. Erepsin. C. Lipase. D. Maltase. And the answer is A. Lactase. Inactive form of the enzyme trypsin, produced by the pancreas and activated by another enzyme, enterokinase, in the small intestine. Its optimum pH is slightly basic. A. Amylase. B. Protease. C. Pepsinogen. D. Renin. And the answer is C. Pepsinogen. An enzyme produced in the stomach, pancreas, and small intestine, which converts lipids to fatty acids and glycerol during the process of digestion. Its optimum pH is slightly basic in the pancreas and small intestine, but acidic in the stomach. A. Amylase. B. Lipase. C. Pepsinogen. D. Renin. And the answer is B. Lipase. An enzyme produced by small intestine that converts proteins to peptides during the process of digestion. Its optimum pH is slightly basic. A. Maltase. B. Protease. C. Trypsin. D. Pepsin. And the answer is C. Trypsin. Pertaining to a group of enzymes that break down lipids. A. Amylolytic. B. Lipolytic. C. Trypsin. D. Lytic enzyme. And the answer is B. Lipolytic. An enzyme produced by the small intestine that converts sucrose to glucose and fructose during the process of digestion. A. Lactase. B. Lipase. C. Sucrase. D. Protease. And the answer is C. Sucrase. A protein digesting enzyme that breaks down cell membranes, found in sweat, acts as a bactericide agent. A. Lactase. B. Protease. C. Lysosome. D. Maltase. And the answer is C. Lysozyme. The amount of the substance to be acted upon by an enzyme. A. Bioreactor. B. Substrate concentration. C. Catalyst. D. Specific. And the answer is B. Substrate concentration. An enzyme that brings about the destruction of cells. A. Amylolytic. B. Catalyst. C. Lipolytic. D. Lytic enzyme. And the answer is D. Lytic enzyme. The substance on which an enzyme acts to produce the product. A. Active site. B. Substrate. C. Enzymes. D. Catalyst. And the answer is B. Substrate. One enzyme will catalyze only one reaction, i.e., enzymes are A. Amylolytic. B. Specific. C. Lipolytic. D. Lytic enzymes. And the answer is B. Specific. Enzyme involved in protein synthesis manufactures messenger RNA from DNA. A. Lactase. B. RNA polymerase. C. Trypsin. D. Maltase. And the answer is B. RNA polymerase. Enzyme produced by the small intestine that converts maltose to glucose during the process of digestion. A. Erepsin. B. Lipase. C. Hydrolase. D. Maltase. And the answer is D. Maltase. An enzyme produced in the stomach when pepsinogen reacts with the hydrochloric acid. It converts protein to polypeptides during the process of digestion. Its optimum pH is acidic. pH is equal about 1 or 2. A. Lipase. B. Pepsin. C. Amylase. D. Maltase. And the answer is B. Pepsin. Enzyme produced in the stomach that converts soluble carcinogen into insoluble casein during the process of digestion. Its optimum pH is acidic. pH is about 1 or 2. A. Erepsin. B. Pepsin. C. Amylase. D. Renin. And the answer is D. Renin. Enzyme produced by the salivary glands which converts starch to maltose during the process of digestion. A. Erepsin. B. Pepsin. C. Amylase. D. Renin. And the answer is C. Amylase. This is an inactive form of pepsin made and stored in cells lining the stomach. A. Erepsin. B. Pepsinogen. C. Renin. D. Trypsin. And the answer is B. Pepsinogen. An enzyme that breaks down proteins, e.g., pepsin and trypsin. A. Erepsin. B. Pepsinogen. C. Renin. D. Protease. And the answer is D. Protease. Best or most favorable pH condition for enzyme activity, etc. A. Erepsin. B. pH optimum. C. Renin. D. Protease. And the answer is B. pH optimum. Hello friends, thank you very much for participating in Enzyme 2. Please do like, share, and subscribe. Thank you. I'm Joshua Mugabi. Bye-bye for now.